All right, so I'm going to go ahead and get started. I think everybody that's planning on being here is here. I'm going to unmute all of you for a second. Maybe. Yes. Those of you that have microphone capability. Can everybody see my PowerPoint?
Yes.
Yes.
Okay, great.
Yes.
I unfortunately cannot disconnect my second screen and still be connected to the right internet, so I may be looking back and forth. I'm just looking at my second screen over here on the side to try to make sure that I'm going through that at the right time. So I'm going to mute you all again, but you're welcome to speak if you would like to unmute yourself. That's not a problem. Just to give you guys a couple of updates, I was able to meet with Raina today. We set up the schedule for the unknown lab. So we are going to start that just the same thing as it was in the syllabus next week. So what is going to happen is that we'll set up the experiments just like if we were running the lab for you. We'll do the inoculations on Tuesday, and then we'll send you video of the results on Thursday. It will be the results of your tube from your unknown. So you'll get an unknown number. There will be, I believe, 6 or 7 potential bacteria. And we'll go through each one of the tests. So we'll go through them just like that was on the schedule. Exercise 34, 35, 36, 37, etc. through the next 2 weeks. And so you'll receive little videos of the results on Thursday from each one of those. And your job will just be to interpret the results and use those results to complete the table in your lab book. So I will also set up an introduction video about the whole unknown project, post the guidelines for the lab report for that. Because you're not actually working in the lab, you are welcome to work in lab groups, meaning that you guys can work together to discuss the results like, hey, I think that's positive, or hey, I think that's negative, in groups of 2. So it can be anybody else from the class. So I'll send out an email after this for you to email me back and let me know if you want to work with somebody to just talk about the results. You do not have to. You can all get your own unknown number. But if you do want to choose a group of 2, then you'll both get the same unknown so that you can work together to figure out what the results are. So I'll send a separate email. You can respond back to me and let me know if you do want to work with someone, and probably someone that you can remotely talk to fairly easily. But you guys will get the same videos, you'll get the same information, you'll get the same pictures. And then at the goal of the end of that is for you to identify what your unknown bacteria is based on those results that you'll get from the videos and the pictures that Raina and I do. So it's like you're doing the lab, but you're not touching the bacteria or inoculating the tubes, but you'll get the pretty videos of the lovely tubes, you'll get the results and try to interpret them. Your grade on that lab report is not based on whether you accurately identify your organism. Sometimes results are a little inconclusive and you have to decide where your results lead you. And it may be that you're picking a bacteria that's not necessarily the correct answer, but if that's where your results lead you, that's all you need to do. So you don't worry about getting the right answer for the unknown. You're just trying to figure out what you have based on the results that you're given. And as most of you know from lab, sometimes things don't work very well, but Raina will give you, and I will give you a picture of what a positive result looks like, a negative result, and then what your unknown is. So you can kind of compare them. Hey, I think it's positive, I think it's negative, and decide on that. Okay. Please feel free to email me with any questions about that after I post the videos, but that again will start next week. You'll get your first video of your number and a plate video of what the culture looks like on a plate on Tuesday. And then you'll get results for, I think it's lab 34 and 35 and possibly some of 36 on Thursday next week. Okay. And I'll go ahead and post the timeline of everything when we're going to get those results to you and when everything is due. So you won't, as far as turning in anything from the lab book, there is no labs reports due for any of that. It's just one assignment at the very end, which will be the lab report. So there's not going to be continuous assignments. It'll just be that very end assignment there. There is a table from the book. Let me pull the lab book real quickly that you'll need to put in with your lab report. And you'll get the results from Raina and I to fill this out. It'll be like a figure in your lab write-up, and that's on page 247, 248. Okay, so that's about the lab report. A little bit more about the schedule for class this week. So I'm planning to do this Zoom session today on chapter 14. I will do a Zoom session on Thursday on chapter 15. Both of those should not go the entire time. And then I will post a link to a YouTube video for chapter 16 that I made actually last year when I was at a conference. So ignore the first part where it says that I'm in Atlanta. I'm not really in Atlanta. I was in Atlanta last year, but it has the information for chapter 16. So you guys can watch that video at your leisure. Still very unknown of what we're going to do about the exams, but as soon as I get the word that we're for sure closing until the end of the semester, I will let you know how we're going to proceed with the exam 3 and then the final exam. We definitely will not have any more quizzes, so don't worry about any quizzes. It'll just be the exam 3 and the final that we'll need to worry about. Okay. All right, so let's go into chapter 14, the bacteria and the archaea. We've been kind of focused on this virus thing, but we do have other microbes that are interesting. Some of the microbes that I will mostly talk about bacteria today. I know that most of you have signed up already for your microbe presentation, but if you after today think, hey, that was a cooler microbe than the one I picked, you are welcome to change your mind. They're not set in stone. So I will talk about some more today. And I will talk a little bit about what we're going to be doing in lab to identify the unknowns, because that's how we classify archaea and bacteria. So let's jump into chapter 14. Maybe. Oh, yes. One more thing before I start. I wanted to do a poll. So I'm going to ask a couple of poll questions today. The first one is totally not related to the lecture, but related to something I may be using for class, which is called Flipgrid. I've never used it before, but I want to know if you guys have used it for other classes before and if it works for you. So here's my poll. Have you ever used Flipgrid before? Yes, I liked it. Yes, it was hard to use. No, I have no idea, or not sure I have. I've never used it. Not sure if I've used it before. Nobody's used it before. All right, so we can all be novices. So what I've been told is that it is a way to post videos and to interact. So I was thinking about using it for the unknown as well as your presentations, but stay tuned for that because I'm not exactly sure. Here, you guys can see. Nobody hasn't used it before. We'll see. We'll see how it works. I've been told it's a pretty good thing to use for video sharing, so we'll see. All right, let's close that poll. All right, so I'll ask a couple of those polls along the way just to make sure that you're still here and you haven't fallen asleep. All right, so bacteria and archaea. If my slide will go forward, let's see, try again. There we go. Okay, so back so many years ago in the medieval times of 1978, a scientist by the name of Carl Woese came up with a classification system to classify organisms. And prior to this, you know, guys like Darwin and other ones that study relationships between organisms had classified organisms based on things that we can see. You know, the phenotype of them, the size of the beak on a bird, or the size of a you know, the fox versus the coyote, things like that, based on morphological characteristics. Well, back in 78, we were kind of delving into this world of sequencing of both DNA and RNA. And Carl Woese at the time thought it was a better way to classify organisms based on their relatedness of their ribosomal RNA sequence. So looking at the sequence of the RNA that's involved in the ribosome and seeing and comparing them between organism and organism and organism. So instead of looking at, you know, the color of their fur and how tall they were, he based this classification system on sequence of RNA or DNA that encodes the ribosomal RNA. Okay? So he decided that life should be classified into 3 domains. The eukarya, and that includes animals, plants, fungal, us. So all the eukaryotes would be classified in eukarya. Then the second domain would be the domain bacteria. And then the third would be the archaea. So 3 domains of life, and these archaea are like our extremophiles, or the ones that live in high salt, high temperature, methanogens live methane. So that was his classification scheme. And this is what it looked like, the 3 domains of life. So we've got the eukarya over here, our archaea, and bacteria. And down here on the bottom is where he found, that's where we talked about this a long time ago, the last common ancestor. So where these all forms of life divulge. Now you can see from this picture that because the tree that leads, kind of the branch that leads to the archaea is closer to the eukaryota here, that based on the ribosomal RNA, the archaea were just as far away from the bacteria, even though those are both considered prokaryotes, than they are from the eukarya. So based on their ribosomal RNA, they look like they were more related to the eukaryotes than they were the bacteria. And this little yellow kind of jelly bean looking bubble is supposed to represent where the microbes fit into that whole domain of life, 3 domains of life. So you can see it, the microbes take up the majority of the organisms that fall into these categories. So I'm going to ask my next poll question. Let's see, domains, launch poll. Okay, so humans are part of which domain of life? Everybody's in agreement that we are a part of the eukarya. We are eukaryotes. Good job, all. Okay. Everybody 100% in agreement. Sounds good. Okay, let's take that off. All right. So yes, we're up here in the eukarya. Ooh, just went backwards. Okay, we're up here under plants, animals, fungi. We're all up here in the top of eukarya. Okay. So this is just kind of a similar branching out of the different organisms. So Luca being, again, a last universal common ancestor, and that each of these domains of life branched off of that last common ancestor. So it kind of coincides with the bottom of the domains there. So the red line down here on the bottom is our archaea, the bacteria up here. And you can see a couple of branch points here. The cyanobacteria, if you guys can remember, cyanobacteria are the first forms of life that produced oxygen from photosynthesis. So they are, they're colored green kind of to remind us that they're very similar to what plants do for photosynthesis. And they were the ones that poisoned the environment with all that lovely oxygen. So the eukaryotic branch here is this blue one that can branches off from archaea down here. And this is supposed to represent our cell and all those lovely organelles. So this kind of being that nucleus, the orange little jelly bean guys being the mitochondria, and then the green guy being the chloroplast. And these little dotted lines represent something called the endosymbiont theory that we'll talk a little bit more about today, where it is believed that a bacteria was engulfed by a eukaryote, so a bacteria that was able to do aerobic respiration, was engulfed by a larger eukaryote and then eventually enslaved as an endosymbiont that later evolved into the mitochondria. And then this green dotted line is another event very similar to that, where a photosynthetic cyanobacteria was also engulfed and enslaved and later evolved into the chloroplast. And we'll talk about a couple of pieces of evidence for that, why we think that's what happened to lead to our mitochondria and our chloroplast that eventually led to our plants here with the chloroplast and the mitochondria, and then the rest of the eukaryote with our mitochondria. Okay. This is a nice little table from another book, just comparing those three domains. So it starts off with the cell type, so the archaea and the bacteria both being the prokaryotes, and then the eukarya being the eukaryotic, the cell wall composition, the difference in the membrane, lipids, the amino acid synthesis. So we can see here in that case that both archaea and eukaryotes use methionine, where the bacteria is using the formal methionine, as we looked at before. RNA loop, we're going to talk about that at the end, the differences of the RNA and the differences of the tRNA down there at the bottom. Okay, so the first classification, when we're talking about the classifications of organism, that hierarchy up there would be those three domains. So the highest level up there on the top is the domain. And then we classify them on each one of these different levels. And you may have seen this before in 121, talking about classification of organisms. Similar things happen in the realms of microbes with the phylum, class, order, family, genus, species, and then specifically we have something in microbes called the strain. We don't normally call like humans having different strain types. We don't call each other strains. So this is something unique to the microbes. So the domain, again, being bacteria. This is E. coli specifically, is in the phylum Proteobacteria, and they're specifically a gamma proteobacterium. Their order is the Enterobacteriaceae. Oh, sorry, the family is the Enterobacteriaceae. Genus is Escherichia, species being Escherichia coli. And then the strain K12 being the one we use in the lab, the kind of benign, everyday E. coli, as opposed to like 0157 that causes our lovely explosive diarrhea. Okay. This gives something similar from another textbook showing the other two domains of life. So here's the eukarya in the kind of bluish color, and this is Saccharomyces cerevisiae that you've all used in lab. That's the baker's yeast. And it just gives you an example of the kingdom, phyla, etc. So one thing you'll notice is that in the realm of archaea and bacteria, we don't have that kingdom classification that you may have seen when you're talking about like 121 classification of organisms in our lovely eukaryotic domain. And then the final classification is always the genus and species, and then strain, depending on if we're talking about, mostly that's in the realm of bacteria. Sometimes in archaea, we are not as adept at growing the archaea in the lab, so they're not as well classified as most of the bacteria that we know about. Okay. Oops. All right, so besides the ribosomal RNA, and the tRNAs that we'll talk about in the end, the other way that we classify microorganisms are based on the things that Raina and I are going to do in the lab for you guys to identify your unknown microbes. And that's by looking at them morphologically. So if you look at them growing on the Petri dish, what does their colony look like? Is it a lovely kind of star shape or a flower shape? Is it nice and smooth? Is it very spready? So we can look at them on the actual Petri dish. Does it have a color? And distinguish them based on those kind of characteristics, which we call morphological characteristics. So how does it look morphologically? Also based on, is it a cocci? Is it a bacillus? Is it a spirocate? So we use those kind of how do they look classifications to classify the bacteria. The second one being the staining procedures. So all the stains you guys did before, spore stain, gram stain, simple stain, negative stain. Raina and I will do that all on your unknowns. And the very most important of those is the gram stain. So being able to first classify your bacteria, whether it's gram positive or gram negative, is really important, because if you're going down the wrong road, pretty much all of your other tests will be meaningless if you don't get it gram positive or gram negative. So Raina and I will do our best to give you accurate representations of each one of your unknowns so that you don't go down the wrong path of gram negative versus gram positive. Okay, so those are differential stains. So how do they look underneath the microscope, and how do they stain gram positive, gram negative? Do they have spores? Are they acid fast? Things like that. And then what the majority of your unknown tests are going to be biochemical tests. So what kind of enzymes do they have in metabolism? What kind of metabolism do they do? Do they ferment? If they ferment, what type of fermentation do they do? There's lots of different types. So we're going to revisit a little bit of what we talked about in metabolism when we're looking at those biochemical tests. So this is just an example of very similar to what you guys are going to get from your unknowns. This is an example of a lot of different bacteria we won't actually be using in the lab. So yeah, a lot of those are pathogenic, so we won't be using very many of those in the lab. Okay, so for the first thing we're going to do for our unknown is the same thing they did on the top here, is we're going to do a gram stain. So if your bacteria came out as gram negative, then you would follow this arrow and go down the path on the left-hand side, versus if it's gram positive, and then on the right-hand side here. And this one ends with the first thing on the gram positive. We will have more tests than this if it's gram positive. So this is just an example. So one of the first tests that we will also do is whether it has the enzyme oxidase. So is it oxidase positive or not? And then if it's oxidase positive, we go down this route down here on the side. If it's negative, we go this. We'll use a test called, does it hydrolyze? Does it cut urea? Does it have the enzyme urease? And that will be used to classify it. Does it use a citrate in the citric acid cycle? We will be doing that test. We will also test for production of sulfite gas, which is on here, or indole. And we'll talk about each one of those tests as Raina and I present the results to you of what those mean. If we get to do a lab practical, that's kind of the things that will be on the lab practical. If we have to do an alternate assignment, we'll talk about what will be on that. But if we ever get to do a lab practical, it'll be answering questions about like, what does indole production mean? So just keep that in the back of the mind. We'll have some sort of test on those, not sure exactly what it will be. But what this allows us to do is classify which bacteria it is. So depending on the answers to this test, we can say, well, this is Klebsiella pneumoniae, or it's Yersinia triclitica, etc. And then we'll do lots of fermentation tests. Does it ferment lactose? Does it ferment glucose? Yes or no. Can it ferment sorbitol? I don't think we actually do sorbitol. I think we just do galactose, glucose, and lactose, I think, are three that we use. Do they generate acid? Other things like that. So this allows us to identify an unknown bacteria based on their reactions to these tests. So if it's positive, leads us down one route. If it's negative, it leads us down the other route. Now it's become more and more easy, instead of doing these biochemical tests, to just sequence the structure or the organism in question. Just go about and sequence it, compare it to the database. That gives us a much more accurate, if it's in the database, it gives us an accurate identification of that organism. So as sequencing gets easier and easier, we'll probably fall in those categories more and more, as opposed to doing some of these biochemical tests. Some of you may have heard, as far as testing for things, that there is a new test out for the coronavirus that is a quick test, not using that RT-PCR anymore. That's supposed to give us results in 45 minutes as opposed to 2 days. And that will cause the number of cases to go up significantly, because we're going to be able to test them more and more and get those results back much faster than those 2 days. So Same as with testing for viruses, testing for bacteria is getting quicker and quicker as well, where we can just sequence them very quickly, very easily find the answers, as opposed to doing these kind of complicated biochemical tests. All right, so let's talk about who the prokaryotes are. Again, if you see or hear something that you find much more interesting than the microbe you originally picked, you're welcome to change yours. So we're going to talk about who some of the stars of the microbial world, as you could say, because as you can see from this, there are a lot of different bacteria out there. We are sequencing more and more. We are learning more and more about the microbes out there. And we're not going to be able to go through every single microbe out there, but we're just going to look through kind of the more famous is kind of a bad word, but the more famous of the bacteria that are out there, a lot of them that cause disease or are environmentally important. And this gives you kind of the general classifications of the bacteria we're going to talk about. There's lots of different ones like spirochetes. We might not talk about so much of those, but the ones that are the color, those are the ones that have the most members that we know about so far. So those are kind of our focuses, being the proteobacteria one being the largest group of bacteria that we know. This is another another table from another textbook that kind of lays out those same things. So if you look at the domain here, we've got domain bacteria, and we've divided them into gram-positive and gram-negatives. Then we've got the archaea down here in the bottom. And we're going to talk first about these gram-negative guys, and the biggest, largest group up there, the proteobacteria. And if you'll notice that these include guys that you probably have heard about, like Vibrio cholera. Oh, hey, my phone's ringing. Hey, and it says I'm calling myself. Wow, that's kind of cool. All right. That include E. coli, Salmonella, Heliobacter, things like that fall into that category. Okay, so we're going to first talk about those gram-negatives and then the proteobacteria. So they're named proteobacteria after the Greek god Proteus, who could assume many shapes. Oops, let's go back that. These guys are what we call chemoheterotrophic bacteria. And with that, we come to our next poll question. If I can get it working. Okay, so what does it mean to be a chemoheterotroph? Do you remember this? They use organic chemicals as both energy and carbon sources. They use organic chemicals as energy source and a carbon dioxide as a carbon source. What do you think? Missing 4 people. Try to get everybody on there. This is really fun in immunology last night because I had control, so Dr. Thompson got a vote. I don't get to vote on this, but I like stumped him, and it was his own questions that he wrote. I can't believe it. He stumped himself, actually. Okay, one more person on there if you want to vote. All right. So yes, most of you guys got this right. It is using organic chemicals as both energy and carbon sources. So if you think about E. coli as your example, so E. coli is our famous proteobacteria. They can use organic chemicals as both a carbon and energy source. So, all right. Get my poll out of the way. Okay. Oops. Now, why are we going backwards? Go forwards, please. Okay. So they're the largest taxonomic group of bacteria. They are the biggest group out there. And there's 6 classes within the proteobacteria, and they're all given these Greek letters, alpha, beta, gamma, delta, epsilon, and zeta, our newest our newest proteobacteria. Okay. So let's go, we're gonna go through a couple of these. I'm just gonna give you again some of the highlights of them. Don't expect that you need to know every single one of them. These are more to give you some kind of general insights into this class. As we get into our specific microbes that you guys present and then I present, just remembering which class they fall into will be important. Okay, so alpha proteobacteria, these guys are associated with plants. And the most well-studied of these guys are Agrobacterium tumefacium. And these guys have this group makes an association with plants, either beneficial or not so beneficial. And that's where Agrobacterium falls in. It actually causes plant cancer. This is kind of a tumor we call it a crown gall that can occur on plants. So interesting bacteria in the fact that it does cause a cancer type thing, not necessarily the same as what we think of as in humans, but like a giant tumor on plants. And you can often see this even on plants on campus, these big giant masses coming out of the plant caused by this Agrobacterium. Okay. Beta protobacteria, two of the important ones that are in this class are two pathogens. Hopefully you have no experience with either one of this group, but it includes Bordetella, which Bordetella pertussis is the responsible for whooping cough, who thanks to the anti-vaxxers that hopefully will be completely eliminated after this whole coronavirus thing, have made whooping cough come back. Whooping cough was almost eliminated. This is one of the vaccines that you guys get very early on for Bordetella pertussis. The second major pathogen in this group is Neisseria, and that includes Neisseria gonorrhea and meningitidis that causes gonorrhea and meningitis. Hopefully neither one of those are something you've had experience with. Here's Neisseria gonorrhoeae. We'll talk, if nobody picks this guy, I will talk a little bit about him when we talk about specific pathogens in a couple of weeks. But this is, the bacteria is the orange and then the cell membrane here, and the bacteria is convincing the cells to uptake the bacteria into the cells. So if you're interested in gonorrhea, you're welcome. But please no gross pictures in the paper of gonorrhea things, or syphilis, if anybody picks syphilis. I don't need those things in there. Okay. All right. The gamma proteobacteria, as you can see from that very long list, this includes lots of different important bacteria, and you'll recognize some of those names on there. Pseudomonas, which includes Pseudomonas aeruginosa, which is an opportunistic pathogen. Legionella. Legionella is an interesting group. It causes a disease called Legionnaire's disease. And it was identified because at a meeting of the American Legion, there was an outbreak of a kind of unknown disease. I'm not gonna say similar to what we're seeing with coronavirus, because it was very specific to people who had been at a single hotel, and they were all overaged white men, which tend to be the demographic of the Legionnaires. And they all came back home and died of this mysterious kind of pneumonia. And they found that the air conditioning unit at the hotel had been infected with this particular bacteria that they identified and named it after the Legionnaires. They called it Legionella after that disease. So also an interesting one with some interesting history if you are interested in picking that one for your microbe. Haemophilus influenza, that was the bacteria that was originally sequenced. It's got the smallest genome. Well, at the time it was the smallest genome we knew. We now have bacteria that have a smaller genome. And then down at the bottom, the one that's bold, the Enterobacteriaceae, and that's the big group of bacteria that includes Salmonella, E. coli. Actually, I'll go into them, but that's our big, we call them enterics because they live in our intestinal system. So we associate these guys living in feces, poop bacteria. So they inhabit our intestinal tract. Some unique things about them is that they ferment carbohydrates. So our intestinal tract is a really great place to encounter carbohydrates. And it is a mostly anaerobic environment. You can imagine deep in our intestines, there's not very little or no oxygen there. So it's a great place for bacteria that ferment carbohydrates because there's no oxygen available and there's lots of carbohydrates. So it's a good place to live. But they can also, they're facultative anaerobes, and hopefully we remember what that means. And here's my next poll question. You can get that up there. Actually, my last poll question. So if you're feeling like pressure, this is the last one. Okay, what does it mean being a facultative anaerobe? You require oxygen. You can grow with or without oxygen, or you cannot grow around oxygen. Had an extra space in there. Almost everybody, two more. One more. All right. Yeah, so most of you guys got this correct. Facultative anaerobe means you can grow with and without oxygen. That was probably the best answer. So they can live without oxygen within our intestine. Then if they're able to leave our intestinal tract and the intestinal tract of other organisms, then they can grow in the presence of oxygen. So again, E. coli being our kind of prototype of this group. All right. They also have flagella, paratrichous flagellated, meaning that they have multiple flagella throughout their body, their cell. Okay. Enterobacteriaceae include E. coli, so Escherichia, E. coli being our fecal contaminant, causing foodborne disease and urinary tract infections. You can think about how a bacteria that lives in our intestine might get into our urinary tract and how that potentially gets there. You can use your imagination on how that could potentially happen. There's also other organisms, but that's one of the main causes of UTIs if you've ever had a UTI, that or Enterobacter. Salmonella, if you're interested in picking Salmonella as your microbe, it's a very common form of foodborne illness. Unfortunately, in this country, most of our eggs and our chickens do have Salmonella in them. So if you go pick up some chickens or chicken from the grocery store, if you just decided to eat it raw and not cook it, more than likely you will bring Salmonella into your body. One of the really bad things about Salmonella is that it doesn't take very many bacteria to make you sick. We call that the infectious dose. If you just picked up 20 or so of the Salmonella bacteria, that could be enough to make you sick, where as other organisms like E. coli, you'd have to take a nice big giant swig of them as opposed to that few of organisms in order to make you sick. Next week, we'll talk more about invading the host, and we'll talk more about that infectious dose thing. Well, what that means for Salmonella is that it doesn't take very many bacteria to make you sick. So again, if you went to the grocery store, took a nice big hunk out of a raw chicken, more than likely you'd pick up some Salmonella food poisoning. It does also cause another disease called Salmonella typhoid fever, caused by Salmonella typhi. If you've heard about Typhoid Mary was a very famous asymptomatic carrier, meaning that she didn't show any symptoms of the disease, but was carrying Salmonella typhi, and she was a cook. So as she was making food, she was contaminating that food. So everywhere that she worked, people were coming down with typhoid fever. And she was going from house to house as people got sick. And finally, someone realized that it was her that was actually spreading the disease. And nobody believed it at the time because she wasn't showing any sort of symptoms. Shigella is a cause of bacterial dysentery. Klebsiella pneumoniae is a cause of pneumonia. Serratia, we've used that guy in the lab. That's the one that has that lovely red pigment. It is an opportunistic pathogen. So for those of you that would have a compromised immune system, it could potentially cause a an infection in those kind of patients. Proteus has this very cool swarming motility. Do I have a picture of it? Yeah. Here, this is Proteus. It's got all these crazy flagella around it. And if you put it on a Petri dish, it makes these kind of cool concentric circles. We call that swarming. So that's Proteus. Yersinia, we've talked about Yersinia before. Yersinia pestis is the causative agent of the plague that is transmitted by fleas. There's two forms of the plague, one that can be transmitted via droplets and one that has to be transmitted via the fleas. Erwinia falls into plant pathogen, another important species if you're in any sort of agriculture. Enterobacteriaceae, again, that is another cause of, Enterobacter causes urinary tract infections. If you have a UTI, it's probably either E. coli or Enterobacter. Since they're both in the same family, they can be treated with the same antibiotic and can usually very easily get rid of that UTI. Chronobacteria, Bacter, which was discovered in 2007 as a causative agent of meningitis, is found in a variety of foods and environments. All right, so those are our big group of Enterobacteriaceae, which fall into the gamma protobacteria. Again, this is our largest group. Depending on which microbe you pick, you might be into this category. All right. Then the Delta, one of the very interesting members of Delta proteobacteria is Della Vibrio. When I was an undergrad, I worked in a lab that studied Della Vibrio. And what makes Della Vibrio cool is that it's a really tiny bacteria that invades other bacteria. So this orange guy is a Della Vibrio, and he is boring himself into an E. coli bacteria. So he sticks the top part of the cell, drills a hole into E. coli and other gram negatives, and goes into that periplasmic space and starts dividing. So it's a bacteria that's pathogenic to other bacteria. So it's kind of a cool, interesting bacterial species. The myxobacteria, so the myxococcal down here, they're an interesting group in that they make little structures on like if you grew them on a Petri dish, they make structures. So, you know, a colony is kind of just a smooth, it's kind of close to the Petri dish. It's not really raised up. Well, these myxococcus bacteria make structures that are actually raised, like little trees on the Petri dish. So they're elevated above the surface of the Petri dish, and we call those fruiting bodies. Myxococcus xanthus is kind of the normal one we work in the lab, and it just kind of makes like a pimple. It really isn't that interesting. It's kind of like a giant mound on there. The myxobacteria that make kind of these tree structures are a little bit more interesting. They're much more complicated, but they form spores, and those spores are used for spreading that bacteria to other locations. So they'll form this lovely fruiting body, and in that fruitobody is the myxospores, and those myxospores can be released to germinate in other locations. So it's kind of a cool structure, developmental structure in prokaryotes. Epsilon proteobacteria. These are bacteria that have a curved shape, and they are microaerophilic, meaning that they don't need oxygen, but they can be around it. They just need a little bit of oxygen, and that's including Campylobacter and Heliobacter. And Heliobacter pylori is the causative agent of ulcers and stomach cancers. This is H. pylori, and again, the curved shape, they're kind of curved like a comma. Here's the flagella. And just like we saw with Della Vibrio, Heliobacter pylori actually bores into the intestine, into your stomach lining, and can cause ulcers, and in extreme cases can cause cancer. The last group of the proteobacteria are the Zeta proteobacteria, and they just have one species. So it's a very newly discovered group of just one bacteria. All right, I'm gonna go back to my table really quick, just to remind you. Okay, so we were in the gram negatives here. We're in the group proteobacteria, and then the Zeta proteobacteria is so new that it wasn't even in this textbook, but it's in your guys' textbook, and that's the classifications here. So that just has 5, but the Zeta one is a newly discovered one. Okay, so those were all our proteobacteria. And then now we're going to fall into the cyanobacteria group. So just to remind you again, cyanobacteria that we talked about in the beginning, those guys are the ones that produce oxygen. So they can do photosynthesis and they produce oxygen. So this lovely equation here is oxygenic, meaning that it produces oxygen. Oxygenic photosynthesis, so using water plus CO2 and light to make our sugar sources, a little bit of water, and then we release oxygen. So they can fall into a category of bacteria that make a specialized structure called a heterocyst. And that heterocyst can also do something called fixing nitrogen, which means that nitrogen gas that's in the environment can then be converted into a usable form of nitrogen. This is a very important portion of the ecosystem that bacteria, these type of bacteria, can fall into. They are photosynthetic, so they need to be close to light. So those that live in the water will have gas vesicles that can provide them buoyancy, making sure they're close enough to the light. And they can be unicellular, so just one cell, or they can hook up to other cells and become what we call filamentous. So this guy here on my left side, I'm not sure what side it's on yours, the long change here are these filamentous bacteria. And that where it says HET, that's the heterocyst, and that job of that little cell there is to fix nitrogen. All right, so those are our cyanobacteria. Let's leap into our gram-positives. And the way gram-positives are divided into their class is either they have low GC, meaning that in their genomes, the amount of cytosine and guanine is very low, or they have high GC, meaning that their quantities of cytosine and guanine are really high. And it tends to be that the gram-positive with high GC tend to live in the extreme environments. So they have that high GC count to keep their DNA in the double helix structure, because remember, those are the three hydrogen bonds between G and C. So having lots of GC bonds in their genome makes their genome a little bit more stable. The low GC include two important ones that are pathogenic or potentially pathogenic: the Clostridium, like Clostridium botulinum, our lovely botulism, or Bacillus anthracis, like anthrax. Then our high GC bacteria, this is another kind of classification system of those high GC bacteria. We can have, do they make spores, do they not, are they aerobic versus anaerobic? And then we've got a couple of classes on there. Do they fix nitrogen, etc. Okay, then just a little bit into our archaea. We're not going to talk too much about them, and then we'll go ahead and be done for today. So archaea and bacteria are both prokaryotes, but those archaea share some characteristics with those eukaryotes, because remember, on our branch, they're like closer to eukaryotes than they are the prokaryotes. So they have, like I mentioned, the methionine, they're more in close proximity with eukaryotes than they are with prokaryotes. All right, so the first thing that we looked at to compare them between the bacteria and the eukaryotes on a genetic level is looking, I guess a molecular biology level, is looking at their tRNAs, so the structure of their tRNAs. So if you kind of remember, the tRNAs are in this clover-leaf structure. This is giving kind of a cartoon drawing of that clover leaf and comparing, the dotted lines are comparing the different structures of the tRNAs in that classification of organism. So down here is the bacteria, then we've got our tRNAs for the archaea, and then the eukaryotes up here. So the yellow shows the kind of difference between the bacterial tRNA and then the archaea tRNA. And then you can see the addition of a couple of extra structures on there, and then the evolution all the way up to what looks like an R tRNAs. The pictures are kind of exaggerated of that structure, so just keep that in mind. they're making them look like the tRNAs are really way off. It's just an addition of some extra base pairs, giving the tRNAs a little bit different structure. And then the end showing that those archaea are much closer to the structure of the eukaryotic than the prokaryotic tRNA. And the same can be true of the ribosomal RNA. This is just to remind you of that 16S ribosomal RNA that's part of the small subunit of the ribosome. So it's this really complicated RNA structure. It's got all these nice loops. Well, biologists like Carl Woese looked at this very complicated structure and compared them and looked at how we evolved from bacteria all the way up to the eukaryotes based on the changes to the ribosomal RNA structure. So this is from a PNAS paper, and I know it's a little overwhelming, but if you look in the middle, that's kind of the structure of this, rRNA, and they've highlighted a couple of kind of differences in the structure. So one up here, one of these little stem loops here, here, and here. And it's comparing the E. coli version of that to the Saccharomyces cerevisiae, which is that baker's yeast version of that, and how it has changed over the years. So we can see here is the E. coli version. And then in the Saccharomyces cerevisiae, that yeast, we've gotten this change in the structure. So we can look at the different organisms and see where do they fall in the growing structure all the way up to what does human ribosomal RNA look like and how are we related there. So they look at these kind of stem loop structures and see how things have changed, how the sequences have changed that have led to the physical structure of the ribosomal RNA and how that has changed. So we can make those comparisons and see how organisms line up to see how evolutionarily they're related. If we potentially look at just one of those RNA sequences, we can look at the changes from the bacteria to the archaea, the yeast, Drosophila being our fruit flies, all the way up to the Homo sapiens, and seeing how there's a small portion that has been conserved through the years, but that we've gotten from this very small structure here in E. coli, all the way up to the very complicated structure that we have up here in us, in Homo sapiens. And we can see how as the organism has gotten more complicated, we've gotten more complicated in this particular section of the ribosomal RNA. So scientists who study the evolutionary relationships between organisms will look at those specific sequences and say, how have the base pairs changed? How can those mutations have been added as the organism has evolved, right? But still keeping that very similar core structure that is all the way back in E. coli. Couple of extremophiles that we find in the archaea that if you guys are interested in using these as your organisms, the ones that live in the extreme acidic environment, so really, really high acid, or the methanogens or the extreme halophiles, if you are interested in using, doing that bacteria, that square bacteria, archaea, Haloarcula, you're welcome to use that as your organism. It doesn't have to be a bacteria or a virus that causes a disease. You can pick anything that sounds kind of cool. All right, so with that, I'm going to stop sharing my screen. I am going to unmute you all. If you want to ask any questions right now, I'm done with that for the Zoom today. I will post the link on there if you want to watch it again. I know it's very exciting to watch this. If you're so bored, you probably do that like 10 times, and you just keep watching it over and over again. I'm sure you do. I will post that right now. I will send the email to let me know if you want to work with somebody in your group. And then next week, look for the videos to start popping up for your unknown project. I will send out the link for the Zoom on Thursday. We'll be looking at eukaryotes. And then I will post the YouTube link to chapter 16 that you guys can kind of watch at your leisure right now. So if anybody has questions, welcome. I don't see the chat up here. I don't know if it went away. If you guys have questions, you're welcome to ask in the chat. Or you can send me an email later. That's fine. I can respond via email as well. All right. So if nobody has anything, your micro presentations, when will those occur? So my plan is to use that Flipgrid thing once I figure it out, and you guys will post those up there so that I can look at them. That will also be when your paper will be due. And again, if you're working in a group, you only have to submit one presentation and one paper. Okay, so just one of each. I know you guys are going to be in separate places, so if you need to work with me to organize that presentation video, one of the good things about the Flipgrid is that you guys can edit it. I'm not so good at that stuff, but if you are, you can use whatever platform you want. You just need to post the video on there. So I will give you that due date. Once I figure out the schedule, I'm trying to work on that today so that I'll get everything posted on Moodle. Now that I'm figuring out the unknowns, got that whole schedule, and I'll get that all. Okay. All right. And you guys are welcome to email me if you have questions. I will see you on Thursday.